Mm. Okay, good evening. Welcome to the first session of Enzyme Sciences and Technology tutorials. Uh, this is a NPTEL course which is taken by Professor Vishal Trivedi of IIT Guwahati. I am Sanket from the Kusuma School of Biological Sciences, IIT Delhi. We will just wait for some more time, maybe a couple of minutes, and let everybody join, and then we can begin with whatever has happened in this week. Am I audible to all of you? Am I audible? Maybe you can raise your hand or maybe give a like, whatever, give a reaction. Okay, I can't hear anything. Are the slides visible to all of you? No reaction yet. Uh, okay, I think we have sufficient number of people now. Again, welcome to the Enzyme Sciences and Technology course. This is taken by Professor Vishal Trivedi of IIT Guwahati. I am Sanket from Kusuma School of Biological Sciences, IIT Delhi. Before we begin, I hope I'm audible to all of you. I got no responses. Maybe one of you can unmute yourself and tell me. Okay. Maybe raise your hand, give some reaction. Uh, Otherwise, it's very difficult to figure out. Since it's the first class, it's going to be the introductory session. Uh, I would expect just to know what are the audience that I'm dealing with. I would like you all to maybe each one of you can tell about yourself. What is your background? That would make a little bit easy for me to make slides further. So maybe you can start by introducing yourselves. Anybody would like to come forward? I don't want to take names. I'm not sure if you can hear me or not. So it's very difficult to figure out like this. It is supposed to be an interactive session, and I would expect some answers also from you as and when required. Uh, okay, if there's nobody is speaking, I would just assume that you can listen to me. So today is the first session. I'm sorry. First session out of the 12 sessions that we are going to have. It's a 12-week course. And every week on Mondays, we would meet from 6 to 8 p.m. and discuss about that week's content and solve a couple of some questions basically, which will be very similar to the ones that you would get in your assignments. So just to start with about this course, first we need to introduce what enzymes are. So enzymes are basically mostly proteins that are very essential for all life activities. Mainly enzymes are important for catalyzing reactions which are important for producing energy in our body. So multiple levels in case of nucleic acids, like DNA or RNA, nucleic acids replication or transcription, translation, all are dependent upon enzymes, right? Enzymes are very, very important. Even in case of lipid biogenesis, synthesis of lipids, and metabolism in various pathways, we have enzymes, multiple enzymes around. um, More than 3,500 enzymes are present in each in a single eukaryotic cell. But before I move forward, I would like to get a confirmation from any one of you that I'm audible and you can read whatever annotations I'm making. Any kind of reaction would work. Please let me know if I'm audible. Maybe you can write in the chat or something.
Yes, sir.
Okay, great, great. Fine. So essentially, enzymes are like they are biological catalysts. You all know this, I assume. So they will essentially lower the activation energy required for a chemical reaction. Now, when we are talking about chemical reactions, there are again of two types, at least in humans, if we talk about or physiological systems to be more specific. They can be anabolic or catabolic. So I'm sorry, just give me. Yeah. Anabolic or catabolic. So in anabolic processes, there is synthesis of new product. In catabolic processes, there is generation of energy. So this is there. Even for nutrition, there is enzymes are required where there is breakdown of complex molecules. We'll read about different kind of enzymes involved in different stages. Then there is the reproduction. Even in reproduction, where we have the gametes which are formed either male or female. There also we have certain regulators, which are again enzymes, and then they form, they're either male or female, and then they form the zygote, and then the offspring. Everywhere, these are important. Now, what are the requirements of life in an organism? If we talk about it, firstly, we require nutrients. That's very important. So for nutrients in different organisms, they could be, let's say, autotrophic. So autotrophic organisms, they will synthesize their own the molecules themselves. So let's take an example, like in case of plants, they get their carbohydrates, the main source of energy, from um, CO2, right? They can synthesize it, and basically, uh, these are autotrophs. Heterotrophs are there, which has certain other classifications also, which are non-autotrophic. Um, they can, feed upon plants, and they can take those complex energy. They could be parasitic organisms, which heavily rely on another organism and their function. Similarly, what else is required? We require oxygen. So again, any kind of complex food, to break it into and to get energy out of it, we require oxygen. So it's very important. And then, of course, water. So many polymeric compounds, many complexes, they are broken down by using water, right? Like let's say starch. So it's broken into glucose using enzymes like amylase. These are called hydrolases. These are the class of enzymes. We'll talk about it. What are the different classifications? But hydrolases, many enzymes are like this, which require water. So water is again essential. Then we need to maintain a normal body temperature, atmospheric pressure. All these things are very important for the life of an organism to, for sustenance of life of an organism. Similarly, there should be a relatively stable environment. So that maintenance of a relatively stable environment inside a body is usually called homeostasis. So that is the term for it. And there are certain changes also which are allowed. It's not that it's very rigid. So though that is called dynamic equilibrium. So these are certain very important key concepts in terms of life, if being concerned. And in all these processes, somewhere or the other, enzymes are functional. We will talk about it. So let's say this is completely different from any kind of enzymes. So let's say a pathogen enters your body. Um, so, you know, just to remove this pathogen or to limit its growth, you develop fever, which is basically a symptom where you have a elevated body temperature to prevent the growth of a foreign pathogen. So that's how your body functions. It always tries to maintain a proper environment so that your processes can happen uh, in the most optimum way, right? Um, Now talking about physiological systems, so in terms of physiology, what are the important these all things have been discussed. So there are like circulatory system, then muscular system, nervous system, endocrine system, excretory system, digestive system, whatever is there, everywhere enzymes are play a very important role. Like in case of circulatory system, there are certain blood clotting factors. In case of muscular system, there are ATPases which break down ATP and all those kind of things. Similarly, in case of, let's say, digestive system, it will basically break complex molecule into simple ones using some enzyme. There are multiple enzymes for this. So these are the basic fundamentals. Basically, in a physiological system, at every step, everywhere, we require enzymes. Now, talking about the major components, major molecules, or what we call as macromolecules. So they are basically, let's say, carbohydrates, lipids, and proteins. These are mainly, broadly, you have these kind of I mean, you can classify macromolecules in these three heads. So starch, let's say carbohydrates, we have starch and other carbohydrates. It could be lactose and all other things. They are also broken through enzymes. So let's say for in case of starch, we have alpha amylase. Then which produces maltose plus glucose, and it gives you energy basically. In case of lipids, we have lipases, which breaks down lipids into smaller fatty acids. Then, for any questions? Is it clear so far, or are there any questions? Okay, I hope not. Um, After that, we have proteins. So for that, we have, let's say, pepsin, or in a broad class, we can say proteases, which break down proteins into characteristic amino acids. Now, this is the all about it. Coming to the concept of catalysis in 1812, I will just brush these off as soon as possible because all of this have been covered. Russian chemist Kirchhoff, he basically was studying about starch degradation. So he boiled starch at very high temperatures, was giving sugar. And he figured out that if you just add a little bit of HCl with it, this process is happens faster with requirement of a less amount of energy. And the HCl which is used is usually is unused. So HCl is not used as a substrate in the reaction. It is not used up. It just remains unused. So it remains unused, but it aids the reaction basically. So that's the concept of catalysis. We'll talk about it. In 1835, Swedish chemist John Berzelius, he coined the name catalysis. So he was the one who said he he coined this name. Okay. If there are any questions, you can raise your hands and maybe ask. So the discovery of first enzyme. Any questions? Okay. Um, so the first enzyme was discovered by digestion of meat. by gastric juices in a hawk. So that was how it was first discovered. Then even starch was discovered by plants like wheat. So starch was basically digested by plants like wheat. They had the protein component of wheat. They could basically digest starch. So that was another way. So French chemist Anselme Payen was the first to discover the enzyme diastase in 1833, and it was basically diastase was extracted from a malt solution. So the study of enzymes is called enzymology. This subject is pretty popular, um, and we will discuss about different enzymes, how they work, and everything. But before that, let's look at the history. So how this developed. So in 1783, as I told you, so first discovery of enzymes where meat was digested by gastric juice of hawks. In 1814, as I told you, the protein component of wheat would convert starch to sugar. Then in 1830, salivary enzyme ptyalin was discovered. Then the first enzyme was discovered in 1833, which was diastase. We already talked about it. Then in 1894, the lock and key mechanism. So it's a mechanism of action for enzymes. We will discuss about it again in future lectures. So lock and key mechanism was identified in 1894. In 1897, cell-free fermentation was discovered. Then in 1905, we discovered cofactors. These cofactors are basically metals which are very important, like let's say magnesium, manganese, even calcium in some cases. These are very important for the enzyme's function. If they are not present, the enzyme do not function. So in 1926, it was known that most enzymes are proteins. Or I would say at this 1926, it was like all enzymes are proteins. But there are exceptions to it. For example, ribozyme, which is an enzyme which is made up of RNA. But anyway, most of the enzymes, almost 99% of enzymes, are made from proteins. So in 1950, um, immobilization was done. So immobilization of enzymes is important for industrial purposes. So you immobilize the enzyme, the substrate comes in, product goes out, the enzyme remains as it is. So this was what was done in 1951. The sequence of insulin was discovered. Insulin is an important protein to maintain the blood sugar levels. In 1953, the structure of DNA structure was discovered. Very important because DNA is the starting material which forms RNA, which forms proteins and basically different enzymes, right? In 1958, the induced fit model was proposed. And then in 1958 itself, the first protein structure was reported. Again, this is a completely different field. It's basically structural biology where protein structures are determined using different techniques like X-ray crystallography, NMR, and uh, nowadays it's cryo-EM as well. So there are different techniques for protein structure determination. In 1966, the genetic code was discovered. basically how codons are important in an mRNA to produce amino acid sequence. In 1968, very important discovery of restriction enzymes were made. These are very, very important to modulate, to produce genetically modified organisms, basically clones. They can be produced using restriction enzymes, very, very much used in molecular biology. And similarly, 1972, recombinant DNA was first made by Paul Berg. So this happened in 1972. In 1977, DNA sequencing was done. I mean, discovered, I would say. So the Sanger's method was the oldest one, even used till date. Then in 1978, site-directed mutagenesis, basically introducing mutations in your genes, that was discovered. In 1978, also, we had the first recombinant insulin, which could be used to treat diabetes. In 1980, another very important technique, which is PCR, was discovered. Then in 1980, in 1991, directed evolution was theorized. In 1997, BASF lipase was found. It's a process to produce chiral amines. In 2010, transaminase was synthesized for sitagliptin. This is important to treat. I mean, this is used to treat diabetes type 2. So insulin, even this drug, and also in 2020, 9 enzyme cascades were found to produce islatravir. It is used to treat and prevent HIV infection. In 2016, uh, an enzyme was found which could engineered basically, which could form the carbon and silicon bond. So these are some of the history of enzymes. So as I already told you that enzymes are biological catalysts. Now enzymes can be studied in two different ways. One of them is the application study. So in application study, what we do is we perform enzyme engineering. We mutate certain motifs. We look for domains which has specific activities, and we engineer enzymes which will give us the best activity. Another one is the theoretical study, which involves physicochemical studies, and let's say catalytic properties of enzymes. So what does this mean? Whether the enzyme works as a monomer, dimer, how it is, how it is activated, if there are certain other enzymes which are involved, all these kind of things are also studied. So as we already discussed, two main models are there, lock and key model, or I would say hypotheses are there, lock and key hypothesis and the induced fit theory, which basically explains the mechanism of enzyme action, right? Um Now, enzymes, they are present in all living cells. They convert substrates into products. They remain unchanged by the chemical reaction, right? This is also important. They remain unchanged by the chemical reaction. They increase the rate of chemical reaction. And as I already told you, 99% of the enzymes, they are proteins. 1% is, let's say, ribozymes, which are RNA containing enzymes. So The question comes that how do these enzymes work? So as very similar to a catalysis reaction in chemistry, enzymes also work in the same principle. So if I make an graph of energy versus reaction coordinate, let's say if I have substrate A plus B, it gives me a product P plus Q. So in absence of enzyme, there would be a very high activation energy. In a presence of enzyme, the activation energy will be lowered. That's the concept, right? So this is in presence of enzyme, this is in absence of enzyme. So lowering of activation energy is basically uh, the function of an enzyme. What is activation energy? Activation energy is the energy barrier that needs to be crossed for the product, for the substrate to form product. So that's the idea. So in case of enzymes, mostly what they do is they break bonds and form bonds. Uh, on an atomic level. So all of this requires energy. So that's why most enzymes they require energy. Although overall in a reaction energy could be produced or exothermic reaction could be there or endothermic reaction could be there, but energy is required in all uh, chemical reactions for the substrate to form product. Now, if we talk about the properties of enzymes, first is they are made up of protein. Second is they are due to the amino acids which are present in these proteins, um, they provide a specific environment for catalyzing reactions with different types of substrate. So let's say enzyme like hexokinase would only recognize hexo sugars, let's say like glucose, and this specificity comes due to the amino acid composition. So third is there is an active site in an enzyme. It's always there in an enzyme structure. You would see there are small pocket, which is the active site from where the substrate binds essentially. Then product is produced in the end. So that is something that is known. So when a substrate reacts with an enzyme, it forms a product. Um, so if the enzyme catalyzed reaction are faster, obviously because it's catalysis here. And then another important thing that enzymes do, especially in physiological systems, is that they generally a reaction which cannot happen in a physiological system at temperatures like 37 degrees, at pH 7 and 1 atmospheric pressure, reactions which cannot happen in that at this kind of a stage because they require higher amount of energy, those reactions are possible due to the action of enzymes. So enzymes help catalyzing reaction in a mild condition. Fine. They are very specific. So specificity is very important in an enzyme. Specificity, okay. Towards their product and substrate both. Now, enzyme activity can be modulated by a non-substrate molecule. This type of control is called allosteric control. And enzyme amount can be modulated by synthesis or degradation. Okay, these are the different properties. Now coming to the specificity of enzyme. How is an enzyme with substrate? There are different reasons. First is the geometric complementarity. And this is basically the theory behind lock and key model. So if this is an enzyme, let's say it is like this, the substrate will be like this. So it fits together. There is a geometric complementarity. So the in the 3D conformation, it's not exactly, it's just a cartoon, but if you look at the 3D conformation, enzyme and substrate, they fit together in a very good way. That's why they are specific towards their substrate. Then there is electronic complementarity as well. So when we are talking about it, it means the electronic charges which are used. So let's say if we talk about DNA, which is negatively charged, all the enzymes which function on DNA, they will have a positive charge usually. So electronic complementarity is also there. Specific amino acids which interact with the DNA, at least they will have positive charge, if not the entire protein. Then there is stereospecificity as well. So stereospecificity mainly means chirality. So let's say all amino acids which are there in humans, they are L amino acids. But only if you see in a bacteria, they have D amino acids. D and L are basically chiral conformation. So there are enzymes which can function only on D amino acids and not on L amino acids, or vice versa. So those kind of specificity is also there. Before I move forward, I would take feedback. Is it clear so far? Yes or no? No answer? Maybe raise your hand, unmute yourself and speak up. I don't know. Okay, fine, great. Um, Talking about the cofactors which are required for an enzyme, cofactors are essentially, as I told you, these are metals, right? These are metals which are involved behind, which are very important for the enzyme action. For example, magnesium ions are very important in case of polymerases, in case of ligases. Magnesium ions are essential. Similar to cofactors, there are coenzymes, which are basically vitamins. Uh, these vitamins are important for, again, it aids the function of an enzyme. There are like coenzyme A, then there is biocytin, FAD plus, all these are different, coenzymes. And since vitamins are important for the enzymes to function, any nutritional deficiency will alter the enzyme's function. And basically, there are many diseases like the pernicious anemia, which happens due to vitamin B12 deficiency. Then there is pellagra. These are some diseases due to vitamin deficiency. It's happened due to vitamin B3 deficiency. Megaloblastic anemia due to the FAD plus deficiency. And then there is beriberi due to thiamine deficiency. Fine. Now we will talk about enzyme classification. Any questions so far? Okay, I hope there are none. So essentially, there are thousands of enzymes in a single cell, right? And the sixth edition of the International Union of Biochemistry and Molecular Biology they categorized 3196 different enzymes in a single cell. So multiple enzymes are basically involved for cellular functions. That is very well known. Um, so enzyme catalyzes reactions involving different types of substrate, let's say carbohydrate metabolism, which will basically have carbohydrates as a substrate. For example, glycolysis. For example, if you know about glycolysis, glycolysis has 10 steps and each step has one different enzyme. So there are 10 enzymes for 10 steps. Fine. And it is not possible to study each and every enzyme by itself. It's not possible. There are so many enzymes. It's not possible to study each one of them. That's why classification is very necessary. So how is enzymes classified? So very simply, the first basis was to classify them on the type of substrate, right? Or the product that they produce, any of them. But it is not a very widely used convention to classify based on that. The International Union for Biochemistry and Molecular Biology, they have only recommended classification based on the reaction, the type of reaction basically that is catalyzed by them. So, for example, a kinase would add, a kinase, any kind of kinase would basically add a phosphate group to the um, substrate basically. So transferases, they will transfer some group into the substrate. So these are how they are classified. So whenever we study new enzyme, why is this classification important? Because whenever we find some novel enzyme, something that has not been found earlier, we can basically try to figure out based on its function that what class of enzyme it falls into. So based on this, there is basis is covered. There is an enzyme commission that has broadly classified the enzymes into 6 groups. We will talk about each and every group in detail, but uh, they are classified based on the reaction catalyzed. Sorry, catalyzed by the enzyme. Fine. And it does not account for amino acid sequence or the protein structure or modifications like glycosylated enzymes, I mean residues or methylated residues, those kind of things are not being uh, accounted for. And every enzyme has a EC number. EC again stands for enzyme commission. And how is this EC number? How would you understand this EC number? It's basically 4 digits. And if we classify that as A, B, C, and D, so A will give you the class of enzyme. So we will talk about the 6 different classes. So there are six possible 1 to 6. Then B is the subclass. C is the subclass, and D is the subclass. Out of these, B and C, it gives you about information about the type of reaction, and D actually tells you about the substrate. So for if you take an example, enzyme with an EC number 1.1.1.1 is basically oxidoreductase. Oxidoreductases are enzymes which essentially they cause redox reactions, basically reduction, oxidation, those kind of reactions. So uh, Similarly, an enzyme with an EC number this 3.4.11.4 is basically this is a specific uh, enzyme which is tripeptide aminopeptidase. So 3 gives for the class. Class is hydrolases. 4 tells you the subgroup, which hydrolases which work on peptides. 11 is the subclass which cleave off the amino terminal of the polypeptide, and 4 is it works on tripeptides. Okay, so if we talk about the 6 classes, uh, so EC1, the first class is oxidoreductase. EC2, the second class is transferases. EC3, and mind you, these are based on the type of reaction that they catalyze, right? Is hydrolases. EC4 are lyases. EC5 are isomerases. EC6 are ligases. So oxidoreductases, as I already told you, cause redox reactions. Transferases, they transfer particular groups. It could be like multiple groups could be there. So they basically transfer uh, uh, from one substrate to another. So that's what they do. Hydrolases, they require water for degradation of, let's say, an enzyme like I talked about lipases. Lipases are example of hydrolases. Lyases also called this degradation, but they do not require water. In the absence of water, they do it. Isomerases, they cause different structural conformations. Sometimes they change chirality. There are certain mutases also which are involved as isomerases. Then there are ligases which basically join two molecules together. Okay. So if we talk about transferases, so they transfer functional group from one substrate to another. So these functional groups could be methyl group, amino group, acyl group, phospho group. These are the examples. So if we take what are the cofactors which are usually involved? Cofactors are magnesium and manganese. Usually these are the cofactors. If we talk about coenzymes, then coenzyme A is usually involved. Example, if we take kinases, which add phospho group, and then there is other kind of transferases. For example, methyl groups are transferred using methyl transferases. And DNMTs are basically there. DNA methyl transferases, which are important to add like methyl groups to DNA, and which is very important for epigenetic regulation. Um, all these enzymes, they can basically modify protein, DNA, lipids, carbohydrates, all these kind of things. What are the subclasses? So subclasses are like 2.1 is transferring carbon group, one carbon group. 2.2, they will transfer aldehyde or ketones, ketonic group basically. 2.3 are acyl transferase, like amino acyl transferase and those kind of thing. 2.4 are glycosyl transferase. They will basically transfer glycosylated groups. 2.5 are alkyl transferase or aryl transferase, alkyl groups or aromatic groups, which are other than methyl groups. 2.6, it transfers nitrogenous groups. 2.7 phosphorus. 2.8 sulfur containing groups are transferred. 2.9 selenium containing group are transferred. So if we look at subclasses, for example, one carbon is transferred, then in that case, there could be, like, as I told you, 2.1.1 are methyl transferases. Similarly, 2.1.2 are hydroxymethyl transferases. 2.1.3 are carboxyl or carbonyl transferases. 2.1.4 are amino transferases. Fine. Now, if we talk about carbohydrate modification, if we look at glycolysis, for example, if we look at transferases here, for example, glucose is converted to glucose 6-phosphate using the enzyme hexokinase. This is a transferase. It requires ATP as a cofactor. Again, it can also form glucose 1,6-bisphosphate using another ATP. So this is how kinases work. Protein modifications can also happen. So let's say there is some protein, it forms a phosphorylated protein, which is usually in its active form. So kinases work here as well. If there is removal of this phosphate by phosphatases, that is also a transfer reaction. Similarly, protein modification is covered. There is DNA modification. So what happens? Very important are the restriction enzymes, which are used in molecular biology routinely. So the idea behind restriction enzyme is that why are they present in a cellular system is to differentiate between self and non-self. How does that happen? I'll explain that to you. For example, uh, usually the DNA that is present with us are methylated. So DNA of an organism is usually methylated. And the DNA of a pathogen, let's say a virus, they are not methylated. They do not have such machineries. They do not have DNA methyl transferases. So they are unmethylated. So the concept is unmethylated DNAs. Let's say this is how it is, with a particular sequence. And its complementary sequence as well. If it is unmethylated, an enzyme like EcoR1, it's a restriction enzyme, will come in, recognize this sequence, leave it off, and form something like this. Basically, it will break the DNA into two parts. And this property is utilized for cloning and other kind of things. Um, so this DNA break is produced because it's unmethylated. If it is methylated, for example, so if we have the same set of sequence, but the adenine here is methylated, in that case, the enzyme will not be able to recognize this sequence and there would be no cleavage. Fine. Now coming to the other group, EC3, that is hydrolases. What do hydrolases do? It catalyzes reaction by forming two products using water. So water is very important for hydrolases. So let's say there's a compound like there's a substrate AB with water, it and the enzyme which is hydrolase will form something like this, AOH and BH. So it will form two different uh, compounds. So if we take the example of hydrolases, the examples are lipase, amylase, peptidase. Most of these enzymes, they are all or phosphatase also, for example, they require they are hydrolases basically. If we classify hydrolases, then they're classified on which kind of bonds they act upon. So 3.1, for example, is they act on ester bond. 3.2 are glycolases. 3.3 on ether bonds. 3.4 are peptidases on peptide bond. Uh, 3.5, they act on CN bonds other than peptide bonds. So there are certain carbon nitrogen bonds which are not a part of peptide. They are basically broken. 3.6 is acid anhydrides. 3.7, they act upon carbon-carbon bonds. 3.8 on halide bonds. For example, CCl bonds with halogen, they act upon these bonds. 3.9 on phosphorus nitrogen bond. 3.10 on sulfur nitrogen bond. 3.11 on carbon phosphorus bond. 3.12 on sulfur sulfur bonds. So they basically break these bonds using water. 3.13 is on carbon and sulfur bond. So this is all about hydrolases. Then comes lyases. Lyases causes non-hydrolytic degradation. So lyases are basically hydrolases, but they do not require water. Fine. Example are decarboxylation reactions or decarboxylases. Pyruvate dehydrogenase. These are examples. So if we talk about the different classes, again, 4.1. So this is basically EC4. Enzyme commission number is 4. 4.1 is carbon-carbon lyase. 4.2 is carbon oxygen lyase. 4.3 is carbon nitrogen lyase. 4.4 is carbon sulfur lyase. 4.5 is carbon halide lyase. 4.6 is phosphorus oxygen lyases, and there is then 4.99 for other lyases. Then comes EC5, which is isomerases. Isomerases, it causes change within a single molecule. So it causes intramolecular rearrangements. So very examples are like isomerases, mutases. Let's say, for example, phosphohexo isomerase, racemase, like alanine racemase, these kind of enzymes, I mean, are examples of isomerases, right? Isomerases also have classification. So their subclass 5.1 are racemases and epimerases. 5.2, they act on amino acids and derivatives. So if some enzyme which converts, let's say L amino acid to D amino acid, they would fall into this category. 5.3 would be on hydroxy acids. 5. Okay, sorry, 5.1.1. Sorry, these are subclasses. 5.1.2 on hydroxy acids, 5.1.3 on carbohydrates, 5.1.4 on other compounds. Similarly, if we look at 5.2, they cause cis trans conversion. So they are called cis trans isomerases. 5.3 are intramolecular isomerases. 5.4 is intramolecular transferases. Also called mutases. They usually call mutation. Then there are 5.5 intramolecular lyases. And there are other groups as well. Then there is ligation, ligases which cause joining reaction basically. So it can synthesize new bonds like carbon-oxygen bond, carbon-sulfur bond, carbon-nitrogen bonds, or carbon-carbon bonds as well. And they usually require ATP as energy, right? ATP is required as energy. So let's say X plus Y will be joined using ATP to form XY. And ADP will be the byproduct of this reaction. Synthetases are examples of this kind of ligases. If you look at this different subclass, so let's say 6.1 is they form carbon-oxygen bond. 6.2, it forms carbon-sulfur bond. 6.3 forms carbon-nitrogen bond. 6.4 forms carbon-carbon bonds. 6.5 forms phosphoric ester bonds. And 6.6 forms nitrogen and a metal bond. A very specific kind of ligase, which is routinely used in molecular biology, is DNA ligase. So how does it function? To understand that, we need to know how DNA ligase, and basically the structure of DNA first. So DNA has a phosphate backbone. It has a sugar and a nitrogen base. So we are eliminating sugar here because DNA ligases, they essentially repair a nick in a DNA. So whenever there is a break, single stranded break, there's an absence of a phosphate bond that will be repaired using a DNA ligase. So this is how a DNA would look like. These are the nitrogen bases. So the bases could be either A, T, C, or G. We all know that. And these are joined together using phosphate bonds. So this is the phosphate backbone essentially. This is one way of representation of DNA. This is usually this is not how it looks like, but anyway, for our illustration purposes, this is fine. Let's say phosphate is not present here. There is a break. Then there is a phosphate here and phosphate. This is a single stranded break, also called nick. This will be essentially repaired using enzymes which are called ligases. So I'll just draw this very quickly. 1, 2, 3, 4, 5, 6, 7, 1, 3, 4. So it is essentially repaired now. And this is done using the most important ligase which is present and used in cloning experiments, specifically is T4 DNA ligase. It is isolated from T4 bacteriophage. It requires ATP. It gives so the T4 DNA ligase buffer, it contains ATP. Similarly, there is E. coli DNA ligase also, which can also catalyze the same reaction, but it will not require ATP. It will require NAD plus as a cofactor. So this is all about DNA ligases. Then we will talk about the enzyme nomenclature. How are enzymes named? So usually there are some trivial names. Trivial names are basically, they do not give you any information, but they are very popular. For example, trypsin, routinely used in cell culture. It's a peptidase. Basically, it breaks amino acid bonds. But trypsin is known as trypsin. So it's trypsin is an example. Thrombin is another example. Pepsin is an example of trivial names. Based on the substrate also, there could be names. So for example, if maltose is the substrate, the enzyme that will break it down would be maltase. If amylose is the substrate, then the enzyme that will break it down will be amylase. And it could be based on the source from where it is isolated. For example, if it is isolated from papaya, that enzyme is called papain. Must have heard about it. Then there is systemic name. Systemic name has usually two words. The first word is for the substrate, and the second word tells you about the type of reaction. Let's say, for example, lactate dehydrogenase actually uses pyruvate as a substrate and forms lactic acid. Okay. What are the rules for enzyme nomenclature? So let's say if you find a new enzyme, how would you name it? So generally accepted trivial names are fine. So for example, trypsin, it's all known. Everybody knows what trypsin is. So that doesn't need to change. But if there is something new coming up, you cannot name it using some trivial name cannot be given. If substrate forms an ion, basically, for example, acid, then it should end with an 8. For example, there is lactate dehydrogenase, not lactic dehydrogenase or lactic acid dehydrogenase. Common abbreviations can be used, but you cannot form new abbreviations. For example, ATPases, we all know what ATP is. ATPase is very well known, but you cannot form new abbreviations. It is usually discouraged. So number 4 is the enzymes which has two nouns uh, should be hyphenated. For example, glucose 6-phosphate 1 dehydrogenase. So there is hyphen in between everywhere. As soon as, so whenever you find a new enzyme, you might say, name it like, let's say it's a condensing enzyme with the particular pH. But all this should be discontinued as soon as you know what kind of reaction it catalyzes, or you can classify that enzyme basically. When a true acceptor is not known, basically, if you do not know that in physiological system, what is the acceptor of the enzyme, then you write acceptors in parentheses. So let's say succinate, you'll write acceptor oxidoreductase. So you know it's an oxido. It forms basically oxidoreductase class of enzyme. It accepts succinate in your system, but ideally, what does that enzyme use as a substrate is not known. And in case of systemic names, it should have two parts. So as I already told you, the name of the substrate, and then there should be the process, how the enzyme works, or the type of reaction, which should end with an ase. That is ideally the rules for nomenclature. We will now start with the quiz, and this time I require answers actually. So I hope you all understood everything. If that is a yes, then we will move forward. I just briefly skimmed through the topics that were covered in the first week's lecture. Is everything understood by all of you? Yes, no? Okay, great. Um, Fine. The term catalysis was first introduced by Kirchhoff or Johannes Jacob Berzelius, Wilhelm Kuhn, or Dmitry Mendeleev. Anybody would like to answer? Please unmute yourself and speak. It's very difficult to talk to a screen.
John's Jacob Berzelius.
You're absolutely right. Absolutely right. So we've already discussed about it. The technique that was used by John Kendrew and his team to determine the first protein structure in 1958 was A, electron microscopy, B, nuclear. Okay, wait. Go to application. Just give me one moment. Technique that was utilized. Wait, I'm waiting for the screen to be visible. Possibly due to charging issue. Sometimes there are issues with IIT's network. I think this time it will be visible. Technique that was used by John Kendrew and his team to determine the first protein structure in 1958 was electron microscopy or nuclear magnetic resonance, X-ray crystallography, or mass spectrometry. Anybody would like to answer? No answers?
Sir, maybe X-ray crystallography.
Yes, you're right. You're absolutely right. The drug was that was produced from 9-enzyme cascade in 2020. We again discussed this. What was it? Was it islatravir, atazanavir, zidovudine, or abacavir? Any answers? No answers? Sorry, I couldn't hear you.
First one.
Yeah, islatravir, it's a drug used to treat HIV infections and even prevent them. The specific location on an enzyme where a substrate binds and the catalytic reaction occurs is called the active zone, reaction center, active site, or binding region. Any answers?
Active site.
Absolutely correct. The specificity of enzymes towards substrate and products is due to the temperature conditions, geometric complementarity, pH fluctuations, or allosteric regulations.
Sir, allosteric regulations.
Uh, okay. Okay. Any more answers? Why allosteric regulation do
Geometric
you think?
complement.
Geometric
Geometric
complement.
complement.
Yeah, yes. Actually, allosteric regulation will modulate the activity of an enzyme, not regulates its specificity. An enzyme will always remain specific towards its substrate. However, the activity of the enzyme can be reduced or increased using allosteric regulation. Is that clear?
Okay, sir. Yes, sir.
Okay, great. The role of nicotinamide adenine dinucleotide is an enzymatic reaction is to transfer acyl groups, or it is to carry high energy phosphate bonds, stabilize DNA structure, or act as a carrier for electrons and hydrogen ions. This is essentially general knowledge. You must have read about it in
Act as a carrier, electron and hydrogen
for
ion.
electrons and hydrogen ions. Absolutely true. In electron transport chain, in even in case of what is it called? Photosynthesis, it acts as a electron transport. Basically, it carries electrons. Coenzyme A functions as a carrier for A, acyl groups, B, high energy phosphate bonds, C, electrons and hydrogen ions, or D, DNA molecules.
Acyl group.
Absolutely correct.
Sir,
Yes.
acyl group, sir. Yes. Okay.
Okay. This is the question number 8. Match the coenzymes with their respective nutritional deficiency and choose the correct option. Hymine pyrophosphate, NAD plus, NADP plus, tetrahydrofolate, and coenzyme B12. And the nutritional deficiencies are pernicious anemia, megaloblastic anemia, pellagra, and beriberi. Again, we have discussed this in today's lecture as well. What would be the option? A, B, C, or D?
Answer 4.
Sorry.
DD.
D.
Yeah,
D.
D is the right answer, where thymine pyrophosphate deficiency will cause beriberi, and NAD plus NADP plus deficiency would cause pellagra. Tetrahydrofolate deficiency will cause megaloblastic anemia, and coenzyme B12, vitamin B12 deficiency will cause pernicious anemia. The information provided by EC number about an enzyme is its genetic sequence, its specific catalytic function, its structural conformation, or its cellular location. Any answers?
Its specific catalytic function.
Absolutely right. EC number is enzyme commission number, if you do not remember. So as we told you, the 6 classes of enzymes, they are all classified and they are given a number based on this. Question number 10. The class of enzymes in the EC system responsible for joining two molecules using energy from ATP is AEC6, ligases or synthetases, EC3 hydrolases, EC2 transferases, or EC1 oxidoreductases.
Sir, E6 ligases or synthetases.
Yes, correct. Very right. An example of an enzyme represented by EC 2.7.1.1 in the EC system is an alcohol dehydrogenase. Is it a kinase, peptidase, or a ligase? Any answers?
Kinase.
Kinase.
Correct. So EC2 is basically transferases. Um, so yeah, the biological process where cytochrome C oxidase, a type of oxidoreductase, participate is glycolysis, DNA replication, citric acid cycle, or electron transport chain during cellular respiration. Cytochrome A, B, C, where are these present usually? No answers? I would wait for any answer to come up. Okay, so the answer would be electron transport chain during cellular respiration. Cytochrome C oxidase is an enzyme involved there. The type of name used for lyase is when the reverse reaction is more important or the only one demonstrated is synthase, synthetase, hydrolase, or reductase. Synthase would be the answer. Question number 14. The isomerase that catalyzes intramolecular rearrangement involving the transfer of functional group within the same molecule is called racemase, epimerase, cis transisomerase, or mutase. We have discussed this.
Racemase.
Not really. It is mutase.
Yes.
So intramolecular transferases are mutases. Processes that are catalyzed by ligases in cellular functions are breakdown of RNA, formation of phosphodiester bonds in DNA, synthesis of ATP, or hydrolysis of proteins.
B. Formation of phosphodiesterase ester bond in DNA.
You're absolutely right. Absolutely right. So with that, I think our questions end here. Any questions from your side? Any feedback? Anything that you would like to ask? Okay. Since this is the first session, I'm really interested to know all of you. Please introduce yourself. Tell me a little bit about your background, what you are doing right now. This will be helpful for me to understand the audience and prepare lecture slides. I mean, to what extent am I supposed to explain? I would understand. So, would you please introduce yourself? Where are you from and what you are currently doing?
Sir, I am Dr. Shanmugh Sundaram from
Okay.
Tamil Nadu, doing assistant professor.
Okay. Okay. In which university?
Actually, I'm working in a private institution.
Hmm. Great.
Uh,
That's interesting. Where did you do your PhD from?
Annamala University, sir.
Yes, yes, Tamil Nadu. Um, okay. Then anybody else? Can see names. Deepali. No answers. Dishant.
Hello, I am
Ah,
Gayatri Dhand and I am from Mumbai. I am currently pursuing BSc in biotechnology.
BS in biotechnology. Which is the university or college?
Bhavans College, Andheri.
Oh, okay, great, great. Okay. Manasvi.
Hello, sir. I'm Manasvi Patil from Mumbai,
Okay.
doing BSc in biotechnology.
From the same.
Currently, yes, sir.
Okay, great. So your batchmates, I think. Mithil.
So good evening, sir.
Uh,
So myself Mithil from Chennai, sir.
okay.
Yes, sir.
And what are you pursuing right now?
I'm doing my master's in drug discovery.
Okay. From where?
Currently, I'm studying in Ramchandra University, sir.
Okay. Okay, great.
Yes, sir.
Uh,
Thank you, sir.
Radhika Sharma.
Good evening, sir. I'm also
Yes.
doing a BSc in biotechnology from Mumbai, Andheri Bhavans College.
Okay. Uh, Sharada.
Hello, I am doing BSMS integrated from ICER TVM.
Aisar Thiruvananthapuram.
Yes.
Okay, great. Shreya.
Good evening, sir.
Hmm.
I'm Shreya and I'm doing BSc in Biotechnology from Bangalore and I'm
University.
from Ramaya University.
Okay, great. Surya.
Hello, sir.
Hmm.
I'm also a second year BSMS student from ICER TVM.
Okay. What is this BSMS in? What is specialization?
Yeah, I am a biology major.
Okay.
Our major started from this semester, like fourth semester.
Cool, cool. Great. Uh, Yash.
Hello, sir. Good evening.
Yes, good
My name
evening.
is Yash Kumar. I am a second year student pursuing BSc Biotechnology from same Bhavans College, Andheri.
Okay, cool. Great. So I think this is how it is going to be. We will discuss the lectures of the that particular week, and then we will discuss the questions and answers. You can definitely going through the lectures taken by Professor Vishal, and if there are any doubts, please bring them forward. This is the time that you would interact with us. To tell about myself, I'm currently pursuing my PhD. I'm in my third year, and I my supervisor is Professor Vivekanandan Perumal. He is currently the professor and head of Kusuma School of Biological Sciences. I also have a co-supervisor, Professor Manoj B. Menon. So I work with viruses mostly. Molecular virology is the field, and what I essentially study is the dinucleotide, the evolution of RNA viruses. So it was great interacting with all of you. I hope from next sessions it would be more of an interaction, and definitely I would be the one who would be talking mostly, but I would also expect that you also would be coming up with certain insights. Not all questions that I would probably know, but I can definitely find out and maybe help you out with it. So it was great interacting with you, all of you. If there are no further questions, I would call it a session now, and we will meet next Monday right at the same time, 6 to 8 p.m. So with that, thank you so much for joining this session.
Thank you,
Thank
sir.
you.
Okay. Great.